Hi everyone, welcome to the BioConference Live. My name is Mei Kong and I'm Assistant Professor from Beckman Research Institute at City Hope Cancer Center in Duarte, California. My lab studies how cancer cells survive metabolic stress. And today I'm going to talk a story focused on protein phosphatase 2A in regulating cell survival upon metabolic stress. So tumor cells often display fundamental changes in metabolism. Compared to normal cells, in order to divide, tumor cells need to take up more nutrients from the environment, usually often driven by oncogenic mutations. So on the other hand, as tumor grows, this increased nutrients uptake and the per-tumor blood vessel often lead tumor cells to face a nutrient deprivation condition. And we believe to make the tumorigenesis persist, and tumor cells need to gain a strategy to survive this metabolic stress. However, how tumor cells survive metabolic stress is still largely unknown. So to answer that question, we need to understand what are essential nutrients used by tumor cells to support their proliferation and their survival. In 1920s, Warburg published a seminal investigation that compared to normal cells, tumor cells take up lots of more glucose. But instead of convert glucose to the T-cell cycle, and most glucose was converted to lactate. And this was named the Warburg effect. And only in these recent years, it has been demonstrated that this Warburg effect was largely driven by oncogenic mutations, such as PI3 kinase, AKT pathway. And the renewed interest in Warburg effect has led to another interesting investigation that not only glucose, tumor cells also depend on glutamine to support their proliferation and survival. So that's why usually in culture media, we need to put lots of more glutamine to make sure your cell line grow well. So why glutamine is important? Because glutamine can actually convert to glutamate and enter into the T-cell cycle and further support the nucleotide synthesis, lipid synthesis. And glutamine itself as an amino acid is also important for protein synthesis. So this increased glutamine uptake or glutamine metabolism also largely driven by oncogenic mutations such as c-MYC. Because as we know, glutamine is a non-essential amino acid, and most non-proliferating normal cells may not need that much glutamine, and it can make by themselves. So targeting these highly proliferating cancer cells become more promising and may cause less toxicity in patients. So actually, many efforts has been put to inhibit glutamine metabolism as a novel cancer treatment, such as DUN and BPTES. They are glutaminase inhibitors. It has been shown that both of them effectively inhibit tumor growth, both in vitro and in vivo. However, all these drug treatment will induce metabolic stress and the adaptive response. So if a tumor can manage to survive, that means this treatment may not be able to efficiently kill tumor cells. So we really need to understand the survival pathway when glutamine metabolism is inhibited. So to support our hypothesis that this metabolic stress do exist in tumor cells, it was reported over 60 years ago by Dr. Eugene Roberts that compared to other amino acids, glutamine level actually falls to almost undetectable in solid tumors in comparison with the normal tissue. So that says lots of solid tumors do experience low glutamine condition and they need to manage to survive. So using a fibrosarcoma xenograft model, we also further detect the glutamine level in these fibrosarcoma tumors. And we also further separate tumor samples from the core region and the peripheral regions. So after we measure the glutamine level, we found compared to liver or other tissue, actually in tumors, the glutamine level dropped dramatically to only around 0.3 millimole. And when we compare within one tumor, we compare the peripheral and the core region samples. We found in the core region, the glutamine level falls almost to an undetectable level. So this data says again, tumor cells often experience a low glutamine level and they need to gain a strategy to survive. So that says as tumor grow, this increased nutrients uptake and the per tumor blood vessel often lead tumor cells to face a low nutrients condition, especially in the core region of the tumor cells. But how does tumor cell adapt to low glutamine levels is still largely unknown. So in the experiment to screen out the genes involved in cell survival upon stress, we identified a protein named alpha-4. And interestingly, alpha-4 plays an important role in glutamine or nitrogen sensing in yeast. So alpha-4 is evolutionary conserved binding protein phosphatase 2A. And otherwise, not much was. So first, we try to test whether alpha also play a critical role in glutamine sensing pathway in mammalian cells. So we just simply overexpress alpha-4 in mast cells. And we found in the vector control cells, when there's no glutamine, these cells died over time. But when we overexpress alpha-4, these cells survived pretty well. So this says alpha-4 play a pretty critical role in mediating cell survival upon glutamine deprivation. So we then asked whether, because we know alpha-4 is a PP2A binding protein, we then asked whether alpha-4 PP2A complex is regulated by metabolic stress, as this complex is actually deassociated upon metabolic stress in yeast. So we did a co-IP experiments, and we found alpha-4 association with PP2A catalytic subunit dramatically decreased upon either glutamine deprivation or glucose deprivation. So this says again, alpha-4 PP2A complex is regulated by metabolic stress. So to further study this physiological role of alpha-4, we decided to generate our alpha-4 knockout mice. However, when we knock out alpha-4 in yeast cells, there's no colonies. So that's kind of remind us in yeast, when people delete type 42, which is alpha-4 homologous in yeast, there's also no colonies. So since alpha-4 probably is a very essential gene, so we went for a conditional knockout system. So we first tried to delete alpha-4 in T cells by crossing alpha-4 flux male mice with the LCK3 female mice, which expression a T cell specific promoter. And then we look at the effect of alpha-4 deletion in T cells. Surprisingly, we found when we delete alpha-4, there's almost no T cells, and all these leftover cells turns out to be some leakage of alpha-4 deletion. So that says deletion of alpha-4 resulting totally depletion of the T cells. So to make the long story short, using this conditional knockout mice of alpha-4, we found when we delete alpha-4 in thymus, there's no T cells. We also delete alpha-4 in fibroblasts and leading to apoptosis. And same thing when we delete alpha-4 in adipocytes or liver, it's all result in cell death. So then go back to the initial question we asked, how alpha-4 protects cells from glutamine deprivation? So we feel like we need to go back and look at the alpha-4 associated protein phosphatase 2A biology. So actually there's two distinct PP2A complexes exist in cells. One is the PP2A catalytic subunit associated with alpha-4 only. There's no other binding protein in this complex. And the other is the classical PP2A trimeric complex, which involve a scaffold A subunit, a catalytic B subunit, and one of the regulatory B subunit. And so far, there's 16 different genes encode the B subunit. And it is the B subunit which determines the substrate specificity or subcellular localization of PP2A. But the role of alpha-4 binding is still not clear. So then we look at the PP2 activity in this active ABC complex or alpha-4 complex by perform IP with either the scaffold A subunit which bring down all the ABC complex, or IP with alpha-4 which bring down the alpha-4 associated catalytic subunit. Surprisingly, we found all the PP2A phosphatase activity only associated with ABC complex. And in contrast, alpha-4 associated PP2A C subunit is actually inactive. So we also look at the effect of alpha-4 deletion on PP2A activity. So we pull down this C subunit to look the total PP2 activity in cells. And we found upon alpha-4 deletion, this total PP2 activity dramatically decreased by over 70%. So that become a puzzle because we know alpha-4 associated C is actually inactive, and only this trimeric complex is active. But why when we delete alpha-4 and the total PP2 activity dramatically decreased? So there's only one explanation, is that alpha-4 functions through modifying this trimeric ABC complex. So we would like to further test that. So we look at this PP2A or C subunit protein level, and we found upon alpha-4 deletion, both A and C subunit, this protein almost completely disappeared. So this says alpha-4 is an essential protein to maintain PP2A, A and C subunit protein levels. So to make the long story short, we found that this ABC complex is actually not very stable. It's consistently undergo assemble and deassemble. When it gets deassembled, PP2AC subunit is actually persists very promiscuous activity and unstable. So it will undergo ubiquitin-mediated degradation. However, alpha-4 could catch this released C subunit and keep it inactive, and that way will prevent ubiquitin-mediated degradation. So that's the major role of the alpha-4. However, back to our initial investigation, it's still hard to explain how we overexpress alpha-4 and cells are resistant to glutamine deprivation. So to figure out this function, we put a hypothesis here. We think alpha-4 could maintain a catalytic subunit in the cells. However, under stress conditions, such as glutamine deprivation, alpha-4 play a role, can pass its associated C to A or B subunit, and further play a critical role in signaling transduction. So that says, to understand how alpha-4 protects cells from glutamine deprivation, we need to identify the specific B subunit that downstream of alpha-4, which mediate this process. So using a real-time qPCR analysis, we screened all the B subunits upon glutamine deprivation. And interestingly, we found there's only one B subunit is significantly induced, which is PPP2R2A, and the protein name is B55 alpha. So we also compared with other metabolic stress and to see whether B55 alpha is only specifically induced upon glutamine deprivation. So we cultured cells in complete medium or glutamine-free medium or glucose-free medium, leucine or essential amino acid-free or serum-free medium. And we found only upon glutamine deprivation this B55 alpha is induced, but not upon other metabolic stress. So this says B55 alpha may be an important downstream B subunit of alpha-4 that play a role to protect cells from glutamine deprivation. So then we asked whether alpha-4 actually provides the C subunit to B55 alpha upon glutamine deprivation, and that way provide a mechanism to mediate cell survival. So to test that, we performed IP experiments with PP2A subunit to bring down all the B55 alpha and C subunit. So we use cells express a vector or flag alpha-4, and we found upon glutamine deprivation, this B55 alpha containing PP2A complex is enriched. And compared to these overexpressed alpha-4 cells, when there's more alpha-4, it seems this complex is further enriched, means alpha-4 plays a role to pass its associated C subunit to this B55 alpha upon glutamine deprivation. So meanwhile, when we look at this alpha-4C complex, we found upon glutamine deprivation, alpha-4 associated C dramatically increased. Means this C was transferred to A and B55 alpha. And next, we also asked whether B55 alpha is required for alpha-4 mediated cell survival upon glutamine deprivation. So to answer that question, we generated stable knockdown cell lines with shRNA against the B55 alpha. And here we use two different clones, C18 and C20. So as we proposed, we think we found when we knocking down B55 alpha, it dramatically blocks alpha-4 protective effect. So as shown here, when we overexpress alpha-4, again, we show these cells are largely resistant to glutamine deprivation. But when we knocking down B55 alpha, both clones are more sensitive to glutamine depletion, even with overexpression of alpha-4. So then next we asked what's the downstream mechanism of B55 alpha that mediates cell survival upon glutamine deprivation. And so first thing we thought about is P53, which is an important sensor to metabolic stress. So a few years ago, it was reported that under low glucose condition, P53 is phosphorylated via either MPK-mediated pathway or ROS-ATM-mediated mechanism. And this phosphorylated P53 further activate a bunch of metabolic gene or cell cycle arrest gene that mediate cell survival under metabolic stress. And recently, another publication reported under low serine condition, P53 is also activated and mediated cell survival via a P21-dependent mechanism under low serine conditions. So here we asked whether P53 is also important for cell survival under low glutamine condition. So we look at the P53 phosphorylation using vector control cells or flag alpha-4 cells upon glutamine deprivation. So we found in vector control cells, p53 is phosphorylated at 48 hours upon glutamine deprivation. Actually, at this time point, we already start to see cell death. However, in alpha-4 overexpression cells, and we know these cells are very resistant to glutamine deprivation, but meanwhile, we found early activation of p53 as early as 24 hours upon glutamine deprivation. So that means this early activated p53 may contribute to cell survival upon glutamine deprivation. So to test that, we overexpressed alpha-4 in wild-type maps or p53 knockout maps. So again, when we overexpress alpha-4 in wild-type maps, we found these cells are very resistant to glutamine depletion. In contrast, in these p53 knockout maps, even with overexpression of alpha-4, they're still very sensitive to glutamine deprivation. So this data says p53 activation is critical for alpha-4 mediated cell survival upon glutamine deprivation. So next, we asked whether this activation of p53 upon glutamine deprivation is alpha dependent. To answer this question, we used this p55 alpha knocking down clones against the 1820. We found under either non-glutamine or glucose condition, p53 is phosphorylated. However, when there's 5 alpha, this metabolic stress activated p53 is largely blocked. So we also look at the downstream 53 target gene using real-time. We found upon glutamine deprivation, this p53 targets, including SSTRINS2 or CDKN1, which is p21, are largely induced under glutamine-free conditions. However, when we knocking down p55 alpha, this glutamine deprivation-induced activation of p53 is largely blocked, which consistent with the Western blot results. So this data suggests this alpha-4 mediated cell survival is through this p55 alpha containing pp2a complex, which further leading to p53 activation and cell survival. But however, what is the direct mechanism mediated by p55 alpha that leading to p53 activation is still not clear. So to further identify that, we performed IP experiments and followed with protein mass spec to identify this p55 alpha associated protein. And there is one, except this pp2a binding proteins, including A and C subunit, we also identified another binding protein with p55 alpha named EDD. And interestingly, EDD was reported to be a negative regulator of p53. So next, we further confirmed whether it is a real data from the mass spec by Western blot. So we IP'd anti-flag p55 alpha and look at the EDD association. And we further validated this mass spec results. And interestingly, we also found the association between p55 alpha and EDD is increased upon glutamine deprivation, also together with the A subunit and C subunit. So we next look at whether this p53 activation by p55 alpha is actually mediated by EDD, which is a direct substrate of p55 alpha. So to do that, we use this p55 alpha knocking down cells, and we further knocking down EDD using the siRNA. So this is a little bit complicated Western blot. So we actually found when we deplete glutamine, again, p53 is phosphorylated. And when we knocking down EDD, so p53 phosphorylation is even higher. So confirmed that EDD is actually a negative regulator of p53. And in p55 alpha knocking down cells, so again, we showed this p53 phosphorylation is largely blocked. However, when we knock down EDD in p55 alpha knockdown cells, it seems this phosphorylation of p53 is largely reversed. So this data demonstrated that p55 alpha activates p53 by directly inhibiting EDD, which is a negative regulator of p53. So in summary, what we found is under like a complete medium or normal nutrient conditions, alpha-4 bind C and maintain the pool of C level in cells. And EDD is phosphorylated, which bind to p53, prevent access of p53 kinases and prevent p53 activation. However, under low glutamine conditions, p55 alpha is induced and further promote assembly of a p55 alpha containing pp2a complex. And this complex can further dephosphorylate EDD, so release EDD from p53. So p53 get phosphorylated and further induce the cell cycle arrest and cell survival. So next we asked, what is the upstream mechanism which mediated this p55 alpha induction upon glutamine deprivation? Because we know that glutamine deprivation actually will induce ROS. So we first asked whether this p55 alpha induction upon glutamine deprivation is ROS-dependent. So we add an ROS inhibitor, NAC, to the medium and look at the p55 alpha mRNA level by real-time qPCR or protein level by doing Western blot. And we found when we inhibit ROS, this glutamine deprivation induced p55 alpha is largely blocked. So this data highly suggests the induction of p55 alpha largely depend on cellular ROS levels. So to further confirm that, we also use a cell-permeable glutathione because when glutamine levels are low, it's leading to decreased glutathione level and further induce ROS. So same as the ROS inhibitor, we found adding back cellular permeable glutathione also inhibit PPP2R2A, the B55 alpha mRNA level, and also the protein level. So again, this all support induction of B55 alpha upon glutamine deprivation is ROS dependent. So we also look at whether downstream phosphorylation of P53 is also depend on ROS. And we found low glutamine again induce P53 phosphorylation, but when we add the NAC to the medium, this increased phosphorylation of P53 is blocked, and also this glutathione also inhibit this P53 phosphorylation. So then next question we asked, because we performed most of these experiments in mast cells, but whether this pathway we identified really exists in cancer cells. because it actually was reported AlphaFold is a protein highly overexpressed in many different cancers. So here we further confirmed whether AlphaFold level is higher in cancer lines compared to 3T3 cells we used. So this is 3T3 cells, and we also, the experiments I showed you, we also use these 3T3 cells overexpress AlphaFold. And when we look at the cancer cell lines, we found most of the cancer cell lines have a higher level of AlphaFold, which including the HT1080 is a fibrosarcoma cells, HCT116 is colon cancer, and U251, LN18 glioma cells, and also MD1, MB231, which is a triple negative breast cancer lines. And then we look at the B55 alpha induction upon glutamine deprivation in these cancer cell lines. And we found almost all the cancer cell lines we tested has an increased B55 alpha induction. And also we look at the phosphorylation of P53, and we found upon glutamine deprivation, the P53 phosphorylation all increased in those cancer cell lines. And P53 is actually also activated when we look at the P53 target gene in these cell lines upon glutamine deprivation. So next we ask whether this B55 alpha is required for tumor cell survival upon glutamine deprivation. So here I show you an example. We use the HT1080 fibrosarcoma cells. So we generated a stable knocking down of B55 alpha using shRNA. And then we tested those cells with non-glutamine medium, and we found upon glutamine deprivation, these cells are not very happy. However, when we knocking down B55 alpha, those cells are much more sensitive to glutamine deprivation. So this is a this is a time course of this cell variability over time compared to scramble control shRNA and B55 alpha knocking down shRNAs. So it turns out to be knocking down B55 alpha makes these cells much more sensitive to glutamine deprivation. So then next question we asked, because these cells, when we knocking down B55 alpha, they're more sensitive to glutamine deprivation. So we then want to test whether knocking down B55 alpha also makes these cells more sensitive to glutamine metabolism inhibitors. So here we treat with L-down, which is a glutamine analog, and now turns out to be also a glutaminase inhibitor. So we found down it's with the dosage we used, down actually did not kill too much cancer cells here. But when we knocking down B55 alpha, these cells become much more sensitive to down treatment. So that highly suggests we can knocking down B55 alpha or use some specific inhibitors to inhibit B55 alpha, and together with the glutaminase inhibitor could effectively kill cancer cells. So as I mentioned before, after tumor cells during their growth, they often experiencing the low glutamine level. So if B55 alpha is important for cell survival under low glutamine condition, so that means if we knocking down B55 alpha, it will affect tumor growth. So to test that, we use a B55 alpha stable knocking down fibrosarcoma cells, and we inject into nude mice to look at the xenograft growth. So as we expected, we found compared to the scramble control cells, when there is decreased B55 alpha, the tumor grows much slower, and compared to the size, it's actually pretty significant. So also, as I mentioned before, we also compared within one tumor. Actually, the glutamine level is much lower inside of the tumor in the core region. So then we want to take advantage of this system to look at the in vivo effect, whether the pathway we identified also exists in vivo in tumors. So we dissect tumor peripheral and the core regions. So here we use a bunch of different tumors which generated from a scrambled shRNA fibrosarcoma lines and also P55 knocking down lines. So P is the periphery sample, C is the core samples, which has much less glutamine. And we found in all the tumor we tested in the core sample, which has much less glutamine, the B55 alpha also induced. And P53 is also phosphorylated. And we also test the P53 target. It's also induced in the core regions. And in contrast, when we look at the B55 alpha knocking down lines, and we found P53 phosphorylation largely blocked, it's actually lower in the core region compared to the peripheral regions. So this confirms that this P55 alpha P53 mediated cell survival pathway under low glutamine condition also exists in vivo in tumors. So in summary, what we found is when glutamine is high and ROS is maintained in a low level, and then B is phosphorylated, P53 is inactivated, and cells stay in. However, under glutamine deprivation conditions, it's leading to increased ROS, and that's further mediate the induction of B55 alpha and further promote assembly of a specific PP2A complex containing this B55 alpha subunit. And this complex specifically dephosphorylates a protein EDD, which is a negative regulator of P53, and that promotes P53 phosphorylation and activation. And this largely mediated cell survival under low glutamine conditions. So actually, during the study to ask how cells survive glutamine deprivation, we actually focus on a lot for serine threonine protein phosphatase. And we also learn a lot for how serine threonine protein phosphatase are regulated. Because we all know that reversible protein phosphorylation almost contribute to all the cellular activities. However, different than kinases, how protein phosphatase are regulated is still largely unknown. So human genome actually encodes around 430 substrate-specific serine threonine kinases. But on the contrary, there's only about 30 different serine threonine phosphatases available. So the central question in the phosphatase field is how do serine threonine protein phosphatase acquire specificity in the face of the promiscuous activity of their catalytic subunits? And also whether serine threonine protein phosphatase are also actively regulated by different signals like kinase does. Is still largely unknown. So I think through study this glutamine sensing pathway, we do learn a lot by which protein phosphatase are regulated. So here we try to propose a novel model by which PP2A complexes are regulated in a stress response. So we think upon stress, AlphaFold could promotes its associated C subunit to a specific B subunit. And meanwhile, a specific B subunit is transcriptionally induced upon specific stress. And that way, promote a specific ABC complex assembly, which could contribute to downstream dephosphorylation and stress response. Because to maintain the C subunit, because C subunit has this promiscuous activity, if not being kept in check, it will undergo ubiquitin-mediated degradation. And alpha-4 actually play a critical role to bind any free C subunit, keep it inactive, and that way we'll maintain a pool of C subunit in cells. So we think this is a very interesting model for regulation PP2A complex, and it's worthwhile to test in other contexts of signal transduction pathways that PP2A complex are involved. So in the last slide, I want to thank people in my lab, especially Mike Reed and Wendy Wong, who contribute to this project, and also all the funding support from different agencies. And I would like to take any questions you guys will have.